welcome to all of you. Happy that you can join us. These are interesting times, so therefore we are happy that we can do this kind of information and present this towards you electronically. We are all getting used to these kinds of formats. Therefore, I'm quite happy that we can reiterate that with this seminar on sepsis, what we did some 2 weeks ago with a seminar on COVID-19, SARS-CoV-2 and Corona. The topic today, sepsis, is not so far away from that because, as you will see in the course of this short presentation, sepsis can also occur, of course, as an adjacent phenomenon while having corona infection. So a lot of our sepsis can also be caused by viral infections, of course, and therefore we are not completely leaving the field, but nevertheless today I would not like to put any focus on the corona infection. So therefore you see the layout of this seminar here. First, sepsis as a diagnostic task, then the requirement in point of care diagnostic and laboratory measures, and then as a final step we would like to give you a state-of-the-art and new markers in assay development. Where are we in the moment with the existing markers? Which markers do we need? You will see in the field of sepsis there's a lot of clinical value and clinical issue behind it. So very focused in sepsis is the clinical aspect of the disease, and the laboratory is helping out and is adjacent to that and endorsing the diagnosis. So therefore we have to focus on the clinical status and on the way how patients are treated and are identified firsthand before the laboratory kicks in. First of all, who are we? Invent, our mission is the targeted procurement of human biomaterials. It is a very general expression, but this is definitely how it is. To our understanding, a precise diagnostic tool, a precise assay needs precise samples, specimen, and this is what we do and provide. To give you a short overview, we are, of course, certified according to 9001, but also to 13485 as a manufacturer of diagnostics. We have our own donation center in Hennigsdorf for donors and patients. We have an own manufacturing site for controls and calibrators. We have a department for clinical services where we perform clinical studies in diagnostics only. So, additionally, we have an R&D unit only for IVD. We are now manufacturers of test kits by ourselves, but we are able to develop them to do something like proof of principle, proof of concepts, and also to transfer to production. So we have a clear understanding how diagnostics are working. We have an extensive experience in procuring, handling, processing, storage, and logistics of human biomaterial of any kind. To give you an idea of our size, we are 50 plus highly qualified employees and 50 plus as full-time equivalents. So this is quite a number of people already, and therefore we are quite competent in supplying any kind of human biomaterial. To meet the demands, on the lower right you see a picture of our donation center. Usually an inquiry comes to invent. Either we have samples or the specimen in our biorepository, small volumes, large volume, whatever it takes. Yes, we do have it, then it will be picked and the samples will be shipped towards the customer, whoever it is, R&D unit, an academic institution, a production, a quality control, proficiency test manufacturers, whoever it is. Or we don't have it, then we start a targeted procurement project to supply the material in time and according to specification, and then shipment takes place a little bit later. We can procure any kind of human biomaterials, so not restricted to serum, plasma, or blood. We can do large volumes and pools up to thousands and hundreds and thousands of liters, normal/healthy and disease state material, as well as clinically defined specimen with low volume but a lot of clinical definition. We put together cohorts and panels, and this is what we can do and provide. So, entering into the topic of today, sepsis as a task. It is a very severe disease. So sepsis, according to the newest definition that there is in the competent world, is defined as a life-threatening organ dysfunction caused by the dysregulation of the host response towards the infection. And this in the past years has been the main game changer. It is not defined anymore as an infection, either bacterial, fungal, or viral, but it is defined as the dysregulated response of the host towards an infection. So that is the disease, and that has been the biggest change in the now valid consensus guidelines towards this disease. We have to take this into account because that is also immediately limiting and directing all what we can do in terms of diagnostics, laboratory diagnostics. It is the third most common cause of death. The forerunners, of course, are cancer and cardiac diseases, and it has an increasing mortality towards age, towards time of identifying of the disease, and in our society, which has to do something with the global change in average age, so the demographic change. It is a multi-organ failure in the end that leads to death, and the classic pathway, which we go in deeper in the next transparency, is infection, sepsis, shock. And then death is the unfortunately quite likely chance, so increasing on this pathway. The mortality risk is increases about 5 to 7% every hour after onset of the disease, before identifying this disease and starting treatment. So time is of the issue. It's a very, very time critical disease, and this is why laboratory diagnostics truly do have a problem if we consider classical diagnostic methods behind it, and then you will see that in a minute how the disease is clinically defined and how the physician enters that. There are more general scores that are at stake here or are involved because they are quicker. The main therapy, of course, not for fungal and for viruses, but most part are bacterial, bacterially caused infections, and then the main therapy is antibiotics. To go a little bit more into detail, we have the infection and the unwanted but quite likely outcome of the disease, death. And if we start that, then there is an infection, then the pathogens enter the bloodstream, so becoming systemic. We have an enhanced production of pro and anti-inflammatory cytokines, so the body is reacting towards that. Then the things are escalating. It comes to a lysis and phagocytosis of the pathogens, and according with that of the endothelial cells, that is then again resulting in the next problem. We have a drop in blood pressure. We have a significant blood clotting because the gaps between the endothelial cells is tried to be closed, and thrombus are forming, and then we have hypoxia because also the fine alveoli in the lung are being blocked. That leads in the end to a multi-organ failure, and multi-organ means more than two is already considered here as an organ failure, and that may result in death if not treated properly and not without good amount of luck in the end. So we start to call the sepsis at that stage on the career of the disease, and this year, starting with the multi-organ failure, is already defined as a septic shock. This has been defined quite recently in the third international consensus definitions for sepsis and septic shock. They are published in 2016. The first have been released in 1991, and then in 2001, both of them are obsolete, again, as mentioned, with the 2016 consensus definitions. The definition has changed towards the away from the infection towards the reaction of the body towards the infection. So a stage further, it follows the, as mentioned, the 2001 guideline, and the definition has been done through a correlation of potential criteria with patient outcome using electronic healthcare data. So how did this consensus group came to the conclusion? As mentioned here, they took a lot of potential criteria, what is defining a sepsis, had tons of electronic data from patient outcomes and patient treatments, and then started a correlation what meets best or is most best to be utilized by the physician to treat the patient, and that has been a multidisciplinary consortium coming up with that. That is the international guideline, and for you, your audience, I would like to show you a little bit more in depth the German guideline, not because to go into detail with them, basically they agree with the international one, but just to tell you why guidelines are so complicated and are treating us or leaving us sometimes with some question marks, how to proceed and how to move on in the field. The medical guidelines in Germany in general, they are an instrument, of course, of quality development in healthcare. They are systematically developed and are a statement to support decision-making of doctors and patients for appropriate care in specific health issues. Therefore, they give a scope for action and decision-making. And so towards the right, you see here the different depths and values. So S1 is just a hint, and then it goes stronger and stronger and stronger. And in S3 guideline, at least in Germany, that's evidence-based and a consensus guideline, and that is almost a law. You have to follow that if you treat a patient. The criticisms behind that, and that is the usual tragedy behind those consensus guidelines, they are extremely useful, they are giving a good guideline, but the trade-offs are it takes the consensus process causes a reduced inclusion of relevant treatment steps into the guidelines because everybody has to agree. And if they cannot agree directly, the people are very eager to be involved in the field, then some processes may be skipped and not mentioned anymore. There are conflicts of interests potentially between if a member of the group is author or editor of guidelines and author of papers and publications behind that. So there may be a conflict of interest in that. It may be inconsistent with international guidelines toward the with neighboring countries and the like. So there are more and more guidelines then, and sometimes they are contradicting themselves. So it's not so easy to deal with that. Of course, there is bias of publications because they rely a lot of public information, published data, and it takes a long time before this is developed now, 2 to 5 years at least. So the next idea to update the German guideline is planned. It has a running time, it's within 5 years, so 2023 is supposed to be the next guideline published. We will see how this works. The German S3 level medical guideline for sepsis by itself, that process has been completed in 2018 and again has a lifetime until the end of 2023, and we have to see how this is going. I don't go into detail here, but as you can see, there are many authors, many associations involved, including Sepsis Aid Group as a patient involvement group. So it's also the question, what is the opinion of the patients being treated like that? 34 authors of those 33 medical doctors. There's a short version with just 41 pages, then there's an extended version with 124 pages, and then a guideline review and an evidence report, which is very substantial and very extended. So that tells you that this is not a simple task to produce that, and what I can tell you is that within this guideline, laboratory medicine is only mentioned to a very, very small extent. So it's more or less decoupled the treatment and the first initial scores aside to what we can do in the routine laboratory in the IVD. So the diagnostics have to stand up for themselves and present themselves more clearly. What the international guideline and the German guideline and most of the guidelines in the world are agreeing on is for the sake of the treatment of the patient, one has to have a quick score, and the most important one is the so-called SOFA score, sepsis-related organ failure assessment. And this one, the SOFA score, is used to track a person's status during the stay in an intensive care unit, or at least in a clinical setting, to determine the extent of a person's organ function or rate of failure. The score is based on 6 different subscores, one each for the respiratory, cardiovascular, hepatic, coagulation, renal, and neurological system. So you see them there on the left side, 1 to 6, and they are then translated nervous systems towards the Glasgow Coma scale, which is the scale by itself. Then respiratory systems, the Horowitz index, giving you an information of the ratio of partial pressure of oxygen in blood and therefore the fraction of oxygen in the inhaled air. So telling you something of the function of the lung. Blood clotting is of course platelet concentration, liver is bilirubin, kidney creatinine concentration in serum, and cardiovascular system is the mean arterial blood pressure. We will come back to that later on. So we have 6 different fields where there is each score and information, and 2 of them have to be significant and positive, and then the SOFA score tells you you have a patient suffering from sepsis. And relevant for us here at that point is we have 3 of them are related one or the other way towards laboratory medicine and diagnostics. So it's a platelet count, bilirubin and creatinine concentration, and plus the microbiological testing to identify the causative pathogen. Again, as mentioned, maybe also viral or fungal as a causative pathogen. As this is a little bit complex and intended to be used in the ICU, there is also a smaller version of that, the quick SOFA score, the Q-SOFA, and that is used to identify patients outside the ICU setting in bedside situations with the suspected infections. That is very important. So the physician or the treating person has to have a suspicion that there might be an infection, and then the Q-SOFA can be used to identify potential poor outcome, which may be typical or which is typical to sepsis, and that is very much reduced. What we see here is again respiratory rate, so elevated respiratory rate larger than 22, altered cognition to this Glasgow Coma scale smaller than 15. Or a hypotension with the systolic blood pressure lower than 100. And these, if these 3 are happening, then it is sufficient for a physician having the suspicion of an existing or the potential of an infection to perform next steps, go to the hospital and consider this as a likely sepsis. If we increase then or are in the hospital, then of course there's the septic shock, and the septic shock is also defined. The definition of the septic shock is provided, so it needs to have the basic requirement that the patient has a confirmed source of infection. Then we have 3 criteria. First is the requirement of vasopressors, so to keep up a mean arterial pressure larger than 65. So the need of these vasopressors to keep up that pressure is mandatory at that point. Lactate has to be above 2 millimole per liter or 80 milligrams per deciliter, and the absence of hypovolemia. So meaning the loss of blood pressure is truly due to the effect of vasodilatation and the vasopressors are needed to keep up the blood pressure. So if that is the case, then this is a full-blown septic shock, and there's a significant number of measures to be applied. So that tells us a lot of work is done via scores and scoring systems, and only a little bit is done in the laboratory. So what we need would be requirements in point of care, diagnostic and laboratory measurements. The issue there, of course, is quite clear. Point of care testing, it shall reduce the time of the sample transport. It has to be done close to the bedside. We need fast results, accelerated treatment start. That is helpful for the patient, of course. Biomarkers that are currently already included in suspected sepsis, although not really mentioned in the guidelines, the CRP, PCT, and cresepsin, and lactate. Lactate is mentioned. Topics with BLCT or point of care testing is we have to have the appropriate clinical context to apply them, and of course the performances in question and the limitations and the cost of the assay are of the issue. What is available already, as mentioned, lactate measurements and several time of blast gas analyzers. They have an additional lactate measuring electrode in them. Point of care devices, handheld, are available for blood gas analyzers as well. Point of care for PCT, there are two at least on the market which are quite well known, all resulting from Brahms using the same raw material. There are no independent companies coming up for measuring PCT. One has to acknowledge that the measurement of PCT, although mainly feeding themselves from the same source of reagents, still have a huge variety in the performance and measurement of PCT. So here two are mentioned, the point of care of Brahms, the PCT Direct, and from Radiometer, the AQT90 flex analyzer. Yeah, that is what is available in the moment. And areas of interest, of course, what are the main problems? It is always a problem of microfluidics. As mentioned before, we have a significant problem with the rheological status of the blood at that point. One has to have that in small measures, and it has to work mainly failure-free. The integration of biosensors and biomarker measurement, well, it's been done for PCT and IL-6. And the next thing would be to amplify the sensitivity of the test while using other detection methods to increase the sensitivity. Yeah, what we would need above all is the quick identification of the pressure trends. So where are we now in the markers in assay development? Why do we need biomarkers? There is still no fully validated biomarker that is that is indicative or decisive to have a sepsis, yes or no. It's always depending about the clinical status, but they can help in critical time period. So in the moment we have difficulties to start the initial treatment and the monitoring. What is in the moment still the gold standard for the diagnosis of blood-borne infections is, of course, the pathogen identification by blood cultures. Long time, and takes a long time, and is too long for to be valid and decisive for sepsis. Moment. The solution would be, of course, sepsis biomarker. That is still the holy grail to be found, and not yet there. These are the numbers that are familiar already, or we are familiar with, that are being used in the laboratories. PCT, of course, for sepsis and bacterial infection, CRP and IL-6 for inflammation. IL-6 has not been worked also good. We come to that later on. The soluble TREM, of course, that is upcoming. Cresepsin is the new star working forward. CD64, super lactate is known already, and some others which are more or less known already. I don't want to go into detail with that. Just the PCT story, that is related to bacterial infections, so cutting out viral or fungal sepsis, causes of sepsis. So cresepsin can go up while PCT is still low if it's a viral or fungal infection. Potential antimicrobial monitoring is possible, and indication for the withdrawal of antibiotics. So it has a good role in antibiotic stewardship, which is not the main problem in sepsis, of course. Here, just that you can see how complex this is. So it's a Procalcitonin is a part of pre-procalcitonin, then cut it here, then we have 3 regions. You have to have the whole sequence of the protein. You have to measure that it's 116 amino acids long, and then you see that you have to have the right antibodies to detect this, otherwise you're just being influenced by calcitonin or catecalcine, which you do not want to have. So specificity, technical specificity is very, very important at that point. PCT is quite quick, 1 to 6 hours after infection goes up, but it is slower than presepsin, which is even quicker. It has a half-life time of 20 to 24 hours, so it has a prognostic significance to a certain extent. But the main thing is whether the patient is reacting to the therapy can be seen. It's reliable in early detection. Grading of severity can be done. Differentiation of anemia and bacteria, bacteremia is possible, and it is definitely more accurate than CRP. CRP is the classical one, but it is very unspecific. It is up and regulated on a lot of cases. It is still being done, and it helps a lot. Of course, everything that we can measure helps us, but you have no conclusion on the septic origin of the inflammation. So it's not giving any result on that. It is also up in traumatic situations and others. Presepsin is quite interesting. It's a fragment of the CD14, the cluster of differentiation number 14, the receptor with that. It has interactions with toll-like receptors, and is important in the recognition of the pathogen-associated patterns. So therefore, it is obvious that it has something to do with sepsis. It is quick. It goes up quite nicely, and it goes down quicker than PCT, but studies are not yet there. But I would think that would be the next big thing to come. At that point, interleukin-6, has played a good role, but is going down in significance. The reason for that is the pre-analytical treatment of IL-6 is very, very tricky. It's fragile molecule. It has a central role in inflammatory reaction, so it's not very specific. Specificity is also a big issue. Therefore, I think interleukin-6 will not prevail in the routine diagnostics of sepsis. Then we have others. I go quickly through with them. CD64 not being proven yet. The already mentioned soluble TREM, still also no hardcore clinical evidence yet, but we have some data towards that. So you see there are some things, the older one are also measured for newborns and adults already. There are sufficient data for that, but for instance, for the newer one, like soluble TREM, there are not even yet numbers for newborns, and also here accuracies are a bit low if you look at the area under curve for differentiation. So that is what we have in the moment. Other things are upcoming. So a new idea quite recently then is to focus more on the origin and the content, the context where biomarkers are coming from, the so-called adipokines, a group of endocrine active proteins from fatty tissue. This may play a role. There are, of course, known markers in that, like TNF, leptin, and also IL-6. But this may play a significant role also in sepsis, not yet known, but there's research going on that. And another one, also well-known or about to be well-known marker is pro-adrenomedulin, or now called bioadrenomedulin. And the interesting topic of this adrenomedulin, and again, it's a pro-hormone like procalcitonin. That is the stable version that can be measured. You see the Kaplan-Meier plots here. Differentiation is you have a lactate that is lower than 2 millimole per liter. Then nevertheless, if you have a low bio-ADM titer, then your survival rate is better than with a higher, but this is at lower lactates. And if you go with a high lactate over above 2 millimolar, which is already a septic shock, then the differentiation between low bio-ADM values and high bio-ADM values is quite significant in terms of survival rate. And you see that the time are in measured in weeks. So it's a prognostic marker, predictive marker for the outcome, predictive marker for the outcome. That is quite interesting, and we have to see how that is working in the ICU, and will come up, and I think we will see that in the next years. Okay, so to give you some outcome on what the routine laboratory now is doing, I'm sorry, I have to apologize, the next to transparency are in German. There are, and that is a group of laboratories we work together well with. They, of course, as a routine laboratory, they have a pathway how to follow any samples. They have a diagnostic pathway that they recommend to their cooperating houses, clinics, hospitals, and physicians, what to do in a sepsis and a septic shock immediately and within 3 hours, and that goes on and on until you get a guideline, so-called guideline, how to treat the patient, when to give antibiotics, what to do next, when to withdraw the antibiotics on. That is how typically routine laboratories are thinking by themselves about the potential of a routine laboratory and giving advices towards their respective physicians treating the patient. So, and that is quite a good information, and brings together the newest standard on laboratory support for deciding how to treat a patient with the suspect of having a sepsis. So further discussions, do we really need new markers in sepsis diagnostics? The answer is obviously yes, it is. There is still a huge lack and huge gap, and no one validated marker is available. What we think is absolutely necessary to do is to strengthen the established markers, to have reliable diagnostic, new designs, point of care, closer to the patient, closer to the bedside, robust systems, updates and combinations of proven technologies. So a combination of several markers will help a lot and support from the IVD side the treating physicians, and discover the challenges in daily routine in lab and hospital is, of course, something that has to be targeted and has to be coped with as a challenge. You can use there things like panels of biomarker positive specimen, of clinical samples, sepsis cohorts, which are ready to use, and that are the tools we can provide to make all those 4 bullet points above happen. If you need any solution with that, I have a question on that, or want to have further insights, then discuss with our expert. This is Mrs. Julia Ettlinger for that. And as sepsis is such a prominent field that has a lot to do with the clinical background, yeah, we have to go further with this. New developments for new markers have a huge demand on time and effort, and cost a lot of money. So PCT now took something like 20 years. Presepsin is now on the market for, I think, 7 to 8 years. Has been heavily invested to get those markers in the reality, in the routine, and this is where the markers should go, end up in routine as standard diagnostics. Therefore, what you need is a prospective analytical and clinical studies, especially according to the new IVDR, and this is a field of activity for our Invent Clinical Services, and our expert would be Dr. Levin Günther, so please do not hesitate to address him. And with this and a delay of 3 minutes, for which I apologize, I would like to end this webinar from our side. I thank you for your attention. Take care of yourself, stay healthy, and feel free to ask any kind of questions. We will be open here for at least another hour to answer your questions. Otherwise, you found 2 email addresses, and please contact those people. I hope you found this a little bit enlightening and compromising and getting things together. Where we are in sepsis, we would like to follow this format here a little bit further, and we will always be at your side to help you with your ideas and to solve your problems and helping us all to increase performance of in vitro diagnostics on the market. I thank you very much and wish you a good day. Goodbye.